require an introduction, but nevertheless, let me introduce. Dr. Kastner obtained his A.B. With honors from Princeton University and a Ph.D. and M.D. from the Baylor College of Medicine. After completing his internal medicine residency, he moved to the National Institute of Health in 1985. He is currently the scientific director of the Division of Intramural Research of the National Human Genome Research Institute. Throughout his career at the NIH, Dr. Kastner's research had focused on using genetic and genomic strategies to understand inherited disorders of inflammation. And this is often stimulated by patients with relatively rare disorders seen at the NIH Clinical Center Hospital. This work has provided the detailed molecular explanations for many of these illnesses and provided the conceptual basis for the highly effective targeted therapies and has informed our understanding of more common illnesses. His group also proposed the now widely accepted and overarching concept of autoinflammatory disease to denote disorders of the evolutionarily ancient innate branch of the human immune system. Dr. Kastner has won a number of awards and honors and has been elected member of the US National Academy of Sciences, the National Academy of Medicine, and also the recipient of the Ross Prize in Molecular Medicine in 2019. Thank you, Professor Kastner, for accepting our invitation. The stage is all yours.
Thank you
Thank
so
you, Dr.
much.
Vinod. Thank you, Dr. Vinod. Now we are moving to the scientific session. The speaker is Dr. Dan Kastner, and the session is on rheumatic disease and the human conditions. Over to you, sir.
Thank you so much for that kind introduction. And let me just share my screen here. Let's see here if we can do that. PowerPoint. So can you all see the PowerPoint
it is visible,
slide?
sir. Okay.
Fantastic. So anyway, it is an enormous honor and pleasure to have the opportunity to talk with you today. For me, this morning, for you this evening. And that is really an especial honor that you all would give up your Friday evening, part of your weekend, to hear me talk about some of the research that we have done. So I really thank you so much for this honor that you have offered me. So anyway, as you heard, the title of my talk is Rheumatic Disease and the Human Condition. And I guess the first thing that I need to do here is to indicate that I have no disclosures to make, no conflicts of interest, no financial conflicts of interest. And let us move on to the talk itself. So rheumatic disease and the human condition. This is an idea that I thought would be fun to consider, especially when this is a weekend talk. And the question that I'm going to pose to you is rheumatic disease inevitable? Is it inevitable as a defense against microbial pathogens? Is it inevitable as the exaggeration of the range of normal? Is it inevitable as a part of the process of aging? And so to consider these three possibilities, I'm going to talk to you about the recent work of three of the fellows in my lab, or who have recently finished working in my lab. And their work has been published over the last 6 months. Actually, one of the pieces of work was published in August in Nature Immunology. Another was published in the PNAS in June. And the third that you're going to hear about is actually available online from the New England Journal of Medicine, but has not yet been published in print. So hopefully these are things that are interesting because they're relatively current and will allow us to think about this question of the possible inevitability of rheumatic disease. So the first question that we wanted to pose is the question of the role of natural selection in autoinflammatory disease. Now, just for those of you who may not be familiar with autoinflammatory diseases, this is a group of disorders. The autoinflammatory diseases are a group of disorders that we first recognized back in 1999 when we described the disease called TRAPS, TNF receptor associated periodic syndrome. And these are disorders in which patients have seemingly unprovoked episodes of inflammation without the high titer autoantibodies or antigen-specific T cells that one would ordinarily see in autoimmune diseases, and of course without any evidence of ongoing infection. So the autoinflammatory diseases are a group of diseases that are primarily caused by disorders of the innate branch of the immune system. That is to say, the part of the immune system that's mediated by myeloid cells, in which the receptors are germline receptors. They're hardwired into the genome. Whereas the autoimmune diseases, as I think many of you know, the key players in the autoimmune diseases are lymphocytes, and their receptors, of course, somatically mutate and rearrange. So anyway, we were posing this question of the role of natural selection in autoinflammatory disease, and we were focusing on one particular autoinflammatory disease that my research lab has been interested in for many years, and that is the disease familial Mediterranean fever. And the person who was doing this work, remember I mentioned that this is the recent work of fellows from my lab, is Yoan Park, who has now gone back to Korea. He was a fellow working with us from Korea, and he was aided and abetted, so to speak, by Jay Che and Elaine Remmers, staff scientists in my lab, and Daniel Schreiner, who's a staff scientist in Charles Rotimi's lab. And they were interested in this question of why are the carrier frequencies for familial Mediterranean fever so high in multiple Middle Eastern populations. Now, again, just some background information for those of you who don't know about familial Mediterranean fever. FMF is a disease in which patients get relatively short, 1 to 3 day episodes of fever associated with either severe abdominal pain or severe chest pain or arthritis. It's caused by inflammation of the sterile inflammation of the peritoneal cavity or the pleural space or the synovial membranes. And at least some patients will go on actually to develop systemic AA amyloidosis and can die of kidney failure, or did die of kidney failure, before the advent of appropriate treatment for FMF. And we discovered the gene for FMF back in 1997. FMF is recessively inherited, and it's caused by mutations in a gene called MEFV, which encodes a protein called pyrin, which is involved in the regulation of IL-1 production. Pyrin nucleates an inflammasome that is important in the activation of interleukin-1 in response to various bacteria. So the question was, why are there such high carrier frequencies for FMF in multiple Middle Eastern populations? And this table shows you for 4 different populations the frequencies of 3 different mutations in the FMF gene, and then the total carrier frequency in these populations. And so you can see that in the Turkish population, the carrier frequency is, let me go forward again. The carrier frequency is 0.127. Which means that roughly 12.7% of the Turkish population are carriers for some mutation, at least of these 3, some mutation in the FMF gene. And you can see that amongst Armenians, the figure is 11.1%, Arabs 10.7%, non-Ashkenazi Jewish people also 10.7%. Those are extraordinarily high carrier frequencies. Just to put that in some perspective, the carrier frequency for cystic fibrosis, which is the most common lethal recessive disorder in North America, is about 4%. And here we're talking about 10 to 12% or so carrier frequencies in toto for familial Mediterranean fever. And what's more, you can see that it's multiple different mutations that are involved in it, and different mutations in different populations, so that it really would seem like maybe this is something that has been selected for in history, rather than that it's just some sort of a coincidence, a founder effect, as one might say, due to some sort of a population bottleneck somewhere in the history of these populations. So that's what we thought, was that it was probably some sort of a selective advantage. But in order to really demonstrate that, you have to do a little bit more than that. And so what one has to do in order to really prove that there would have been some sort of selective advantage for mutations in the FMF gene is that you have to look at the chunk of DNA surrounding the FMF gene, either in people who are carriers for FMF mutations or in people who don't have FMF. And we happen to have genomic data for actually about 2,500 individuals from Turkey that we had been studying to study the genetics of a different disorder, Behçet's disease. But it allowed us to do this kind of analysis where we had a fairly dense map of genetic markers throughout the genome for this 2,500 or so Turkish individuals. And then we could look to see what is the chunk of DNA that's conserved amongst people who have a particular FMF mutation versus those who do not. And so here you can see for the M694V mutation, which is a very important and severe mutation in the FMF gene, you can see that the chunk of DNA that's associated with the wild type allele is very small. It's just like this spike that you can see here. Whereas the chunk of DNA that's associated with the M694V mutation is very big. It's a very broad chunk of DNA. That means that the DNA fingerprint that's associated with this mutation is a fairly large one, extending 1 million base pairs out on either side, at least in some individuals. So that's the kind of thing that you would expect to see if there was some sort of a selective advantage, because basically that's telling us then that essentially recombination has not happened as rapidly around the mutation as it has around the wild type allele, because of the fact that there has been possibly some selection. But you need to have even more information than that. And that is that you have to compare these haplotypes that are associated with the mutant allele with other haplotypes throughout the genome of Turkish people that would be at the same frequency. And so if you do that, then what you're looking to see is whether the haplotype that is associated with the FMF mutation is, when it's compared with haplotypes of the same frequency, is it at the edge of the curve? Is it at the tail of the bell-shaped curve? And you can see that, in fact, it is at the tail of the bell-shaped curve. So this is definitely telling us that there was selection of that particular allele. And we can do the same thing, and I won't show it to you here in these slides, but in the paper that I will tell you about in a little bit, we have data with regard to the V726A mutation, which is another important FMF-associated mutation. What's more, one can analyze these kinds of genomic data to get some sort of an idea of what kind of selection, what is the magnitude of selection that this M694V mutation might have been undergoing. And there's a way of calculating that, and that is shown basically in the lower left-hand part of this slide. And you can see that the selection intensity is indicated as 0.077. So how does that compare with anything? Is that a high selection coefficient or not a very high selection coefficient? Well, so the selection coefficient for lactase persistence, which you know is the ability to digest cow's milk, and that's something that's been under selection in the Northern European population and is a sort of a classic example of a trait that has been under selection, is shown here at the bottom here as 0.056. So a selection coefficient of 0.077, that's high. That, again, is really telling us that these mutations, or this particular mutation anyway, in the FMF gene, has been under selection, strong selection. And then finally, one can estimate when that mutation would have first arisen and then been propagated into the Turkish population anyway. That's what we're looking at here, is specifically relevant to the Turkish population. So there are three different methods one can use to estimate the age of the mutation, and you can see that the age of the mutation ranges from 88 before the Common Era, before Christ, to 3,500 years before the Common Era. In other words, the mutation is somewhere between 2 and, say, 5,000 years old. So that's about the kind of range that you get when you do these kinds of calculations. It's not like it will tell you the exact year that the mutation arose, but it is telling us that the mutation would have arisen after the onset of civilization and agriculture in the human population. So at least relatively recently, although not real recently in human history. So it gives us an idea that the mutation is somewhere around 2,000 to 5,000 years old. So we then have evidence that FMF mutations have, in fact, been under strong selection in the Turkish population, and that these mutations probably arose between 2,000 and 5,000 years ago. And we have similar data for the V726A mutation again. So then you would then ask the question, well, so what could it have been that was selecting for the high carrier frequency of FMF? And in order to consider that question, you have to think about, well, what actually does pyrin do in normal physiology? And so here, I'm going to show you on this slide just a schematic diagram of where does pyrin fit into normal biology. So here, this at the bottom is a depiction, a schematic of the pyrin inflammasome, which, as some of you probably know, the pyrin inflammasome, involves the interaction of pyrin itself with a adapter protein called ASC and the protein caspase-1. And when they assemble into a complex, then one can get basically the cleavage of interleukin-1, pro-interleukin-1 beta, to the activation of IL-1. So this pyrin inflammasome can form under some circumstances. Now, ordinarily, the pyrin inflammasome is highly regulated and is inhibited by the binding of a protein called 1433 to pyrin, because pyrin ordinarily is phosphorylated at a couple of sites, 208 and 242. But that phosphorylation is mediated by a protein called PKN, which is actually activated by Rho-A, which is a GTPase that lives inside the cell membrane. So what ordinarily is happening is that Rho-A activates PKN, which causes phosphorylation of pyrin, which then leads to 1433 binding of pyrin, which blocks the pyrin inflammasome. So that ordinarily we're not walking around with our pyrin inflammasomes activated, making a lot of IL-1, because if so, then we would be walking around with fevers all the time and a lot of inflammation. So it's a tightly regulated process. But what triggers actually the activation of the pyrin inflammasome are bacterial toxins that inhibit Rho-A. And when Rho-A is inhibited, then PKN is inhibited, and the phosphorylation of pyrin is inhibited, and 1433 doesn't bind to pyrin, and so the pyrin inflammasome can form. Now, bacteria have, over the ages, developed toxins that actually inactivate Rho-A. Now, the reason that bacteria have evolved toxins that inactivate Rho is that Rho is actually a very important molecule for white blood cells, to do the things that they need to do. White blood cells need Rho to be active in order for them to migrate, in order for them to phagocytose, for them to degranulate. And so bacteria have evolved a bunch of different toxins over the ages to inactivate Rho. And we think that the pyrin inflammasome is in fact sort of a host response, if you will, to these bacterial toxins, so that we have a way of countering bacteria that can inactivate Rho. And so that's the basic story in terms of the pyrin inflammasome. And so then you can think about, well, what bacteria make toxins that inactivate Rho, and what bacteria are so lethal that, and were, epidemic or pandemic in their magnitude, that it could make some sense that they might have selected for the high carrier frequencies of pyrin. And so, and FMF mutations. And so one of the bacteria that can activate the pyrin inflammasome is Yersinia pestis, which is the organism that is responsible for, that causes the bubonic plague. And Yersinia pestis makes two toxins, YAP-E and YAP-T, which inactivate Rho, and so therefore activate the pyrin inflammasome. So, in the interaction between man and bubonic plague makes a toxin that activates the pyrin inflammasome, so we have a defense against the bubonic plague. But more recent versions of Yersinia pestis, versions of it that have been involved in the epidemics that we know have occurred over history, have another toxin called YAP-M. And YAP-M is a diabolical toxin, which actually can cause a protein called RSK2 inside the cell, actually to phosphorylate pyrin. And when that happens, of course, when pyrin gets phosphorylated, then 1433 binds to pyrin, and it blocks the pyrin inflammasome. And so, we have bubonic plague making these toxins that inactivate Rho. The pyrin inflammasome is our response to that, but then bubonic plague has made another toxin that inactivates the pyrin inflammasome. So that's an interesting thing. And what we're going to show in the next couple of slides is that actually mutant pyrin, the pyrin mutants that are associated with FMF, are immune to, if you will, are unresponsive to this YAP-M-induced phosphorylation. And so it's actually a protective thing, which has been selected for. And so here, this slide just shows that this is the phosphorylation of pyrin, either without YAP-M or with YAP-M, induced by PKN. And you can see it's not a whole lot of phosphorylation. But on the other hand, RSK, this other protein that I told you, it really dramatically phosphorylates pyrin. So it is a strong inhibitor of the pyrin inflammasome. Now, the interesting thing, as I mentioned, is that if you actually look then at... I'm sorry, I think that maybe someone is also talking. But anyway, you can see on the left-hand side, these are basically lysates from healthy control individuals that have been exposed either not or to Yersinia pestis, and then looking for phosphorylated pyrin. And you can see, again, that in the healthy controls that Yersinia pestis induces a lot of phosphorylation of pyrin. And you can see that in all of these circled things. But if you take the lysates from patients who have FMF, and the mutations of these people are shown across here, for those 5 individuals, if you look at the phosphorylation of pyrin that's induced by Yersinia pestis, it's not so much at all. In fact, in some cases, there's hardly any phosphorylation of pyrin. So that's what I'm saying, is that the mutant form of pyrin, the FMF-associated mutations, are resistant to YAP-M-induced phosphorylation. And that then translates into increased IL-1 production in vitro. You can see here when we expose either control cells, when we expose either control cells or cells from FMF patients to Yersinia pestis, we get a lot more IL-1 being produced by those FMF cells. What's more, if one looks at just heterozygotes, in other words, carriers for FMF, people that may not have FMF itself, but they're carriers for it. They just have a single mutant allele. And you can see that, again, they produce increased amounts of IL-1 beta in response to Yersinia pestis. However, if you look at a Yersinia pestis that does not have YAP-M, and remember I said that YAP-M was the thing that makes all the difference here, then there is no difference between the control and the FMF cells in terms of the amount of IL-1 being produced. Or if you expose them to Burkholderia senecapacea, which is a bacterium that doesn't have YAP-M, but is similar in some ways to Yersinia pestis, again, you don't see that difference. What's more, if we breed mice that have the FMF mutation knocked into them, we see an interesting phenomenon as well. Now, of course, we wouldn't want to expose humans to Yersinia pestis and see whether or not FMF carriers survived better than control individuals when exposed to the plague. That would obviously be unethical. But one can do at least similar kinds of studies in mice. And so what one can see here, the green line represents mice that carry the wild type form of human pyrin. So there is some protection of those mice versus normal mice that don't have any human pyrin in them. So that's the baseline that we have to use for our comparisons. The red shows mice that have one of the FMF mutations, and you can see that they are protected. This is survival, and they are protected relative to mice that have the wild type human pyrin. What's more, if you breed these mice to a strain of mouse that doesn't have the IL-1 receptor, shown here in blue, then that protective effect goes away. And so that's showing you that the protection induced by the mutant FMF allele is, in fact, a protection that is related to IL-1 production. And then, finally, the question that I'm sure many of you are asking is, well, but could this really explain the high frequencies of FMF mutations that we see in the Turkish population or in other populations? And so, to answer that question, we actually did some simulations based on what we know of when the FMF mutation would have arisen, what we know in terms of when there were pandemics of bubonic plague. And so when there were pandemics of bubonic plague, there was one of them called the Justinian plague that went from about 541 AD, or in the Common Era, 541 to 767. That was the Justinian plague. And then, of course, the Black Death started in 1346, and in the Middle East, actually, the Black Death was relatively prevalent until the mid-1800s. So if you look at what happened in terms of the frequency of the M694V mutation in red and the V726A mutation in blue, you can see that both of them went up during this period of time. This is the Justinian plague that I mentioned right here. And then during the period between the Justinian plague and the Black Death, it just sort of leveled out the frequency of M694V and V726A. And then during the second pandemic, which actually lasted a longer period of time, you can see that the M694V mutation, it went up to a allele frequency of 0.03, which then translates to a carrier frequency of 0.06, and the V726A went to an allele frequency of 0.02, which is a carrier frequency of 0.04. The carrier frequency is twice the allele frequency. And that actually is pretty consistent with what we see in modern day populations in terms of the frequency of FMF mutations. So, in any case, what this is telling us then is that for sure, FMF-associated mutations in pyrin have been under selection over the centuries, and that, at least from a biologic point of view, we have mechanistic data that would suggest bubonic plague could have done that, and that is consistent with the numbers that we actually see in terms of frequencies. If you'd like to read more about this, it came out in the August issue of Nature Immunology, and you can see here the paper. This is the title page of the paper for your further review if you'd like to learn more about this. I'd now like to turn to our second story, which will be perhaps a little bit shorter than this one, just so that we won't keep you up until midnight tonight. And this is a story about the spectrum of mucosal inflammation. And this is a story that stars Kalpana Manthram. Now, Kalpana actually is a former fellow on my lab. She's now a junior faculty person in the National Institute of Allergy and Infectious Diseases. And she's a first-generation American. Her parents actually are from India, from southern India, the Bangalore area. She was aided and abetted. She collaborated with Elaine Remmers and two other former fellows in the lab, Yohei Carino and Masaki Takeuchi. So in any case, this story begins with our interest in a condition called Behçet's disease. And just to remind you with regard to Behçet's disease is not a monogenic disease like FMF, like familial Mediterranean fever. Instead, Behçet's disease is usually a genetically complex disease where there are a number of different common variants that are associated with susceptibility to Behçet's disease. And of course, clinically, Behçet's disease is characterized by this triad of painful oral ulcers, ocular inflammation. This is hypopyon uveitis, where there's an accumulation of pus in the anterior chamber of the eye in a patient, and then genital ulceration. So that combination of three things are important in making the diagnosis of Behçet's disease. And we also have the pathergy phenomenon illustrated here, in which some patients with Behçet's disease will develop pustules on their skin when it's pricked with a sterile needle. Down here at the bottom, it says lambda sub s is greater than 10. What that means is that the probability of a sibling of someone with Behçet's disease getting Behçet's disease themselves, that it's more than tenfold what it would be for someone that's the sibling of a non-Behçet's patient. So there's at least a tenfold increase in risk of having Behçet's disease if you're a sibling of someone who has it, indicating that there are genetic factors, although, as I said, it's not usually Mendelian factors. This slide simply illustrates something that we think about a lot in terms of human diseases. And along the x-axis, we have the allele frequency. Along the y-axis, we have the effect size. And so what I'm saying basically is that Behçet's disease is a disease where there's common variants that have a relatively low effect size. That's the usual situation for Behçet's disease. Whereas FMF would be here, where you have fairly rare variants that have a high effect size. Now, we started working on the genetics of Behçet's disease back in about 2008 or so, and started publishing on it in 2010. And some of the papers that we've written about this are at the bottom of this slide. And of course, it was known before we started working on Behçet's disease that HLA-B51 is associated with susceptibility to Behçet's disease. Using a genome-wide association study design, we were able in 2010 to show that variants of the interleukin-10 locus and the IL-23 receptor locus are associated with susceptibility to Behçet's disease. And then afterwards, we found several other genes that are also associated with Behçet's susceptibility. 3 of them are shown there in this lower right-hand side. And then an additional one that's very important that I'll illustrate to you on the next slide is a gene called ERAP1. There are some sort of intermediate frequency variants, including the FMF gene. The M694V mutation in the FMF gene actually confers susceptibility to Behçet's disease. So there are a bunch of medium frequency variants that are associated with Behçet's susceptibility, and then a whole bunch more that we've found, so that altogether there are 17 different genes that we had found that are associated with Behçet's susceptibility. And there even is a rare monogenic form of Behçet's disease as well, caused by mutations in TNFA-IP3. Just to illustrate a couple of these, and we can't go into detail on all of them. I said HLA-B51 is associated with Behçet's susceptibility. There are some other HLA types that are also associated with Behçet's susceptibility. Mike Ombrello in my lab did that work back in 2014, and what he found was that the key amino acids in the class 1 MHC molecule that confer susceptibility to Behçet's disease are along the binding groove of the class 1 molecule. So it suggests that maybe there would be some sort of an antigen that's being presented by HLA-B51 and other HLA alleles that are associated with Behçet's susceptibility. What we found at about that same time was that there's coding variants in this gene called ERAP1 that are also associated with Behçet's susceptibility. And that's really interesting because ERAP1 encodes a protein that lives in the endoplasmic reticulum and that's involved in trimming peptides and loading them onto class 1 molecules. And it turns out that the variants of ERAP1 that are associated with susceptibility to Behçet's disease only work in people who are HLA-B51 positive. So it really, because of the fact that there's this epistasis, this interaction between these susceptibility alleles, it really does make one think that perhaps there are Behçetogenic peptides that may be associated with the initiation of Behçet's episodes. With regard to the monogenic form of Behçet's disease, just one slide on that. TNFA-IP3 is the gene that's involved there. It encodes a protein called A20, and A20 is a molecule that regulates ubiquitination of various proteins. And ubiquitination is a post-translational modification of proteins, and it can have an important effect on regulating immune responses. So in this particular case, you have a cluster of proteins, a signaling complex, that's induced by tumor necrosis factor that is stabilized by K63 ubiquitin that's linked together at lysine 63. A20 actually is a deubiquitinase for this particular reaction. It removes K63 ubiquitin chains and in that way destabilizes the signaling complex, and the constituent parts of it come apart, and then A20 actually has a second function where it can add K48 ubiquitin chains to the constituent proteins that come apart from these signaling complexes, and that targets them for destruction by the proteasome. So it's a sort of a 1-2 punch, if you will, where A20 is important both for destabilizing inflammatory signaling complexes and then degrading the constituent parts of the signaling complex. So you would then think that if you had a mutation in the gene that encodes A20, a loss of function mutation, that might lead to a hyperinflammatory process, which is in fact what we have seen. So anyway, 3 years ago, we had gotten to this point, and we were very proud of ourselves in terms of having found all of these Behçet's susceptibility loci. And then I went to the American Society of Human Genetics meeting. And that was quite an experience because I went to a talk by someone from 23andMe. You know, 23andMe is this company that does direct-to-consumer DNA testing, so that if you do some sort of a buccal brushing and send a sample to the people at 23andMe, they will do DNA sequencing and tell you a little bit about your ancestry and a little bit about your susceptibility to common diseases like hypertension and cardiovascular disease. But they also, apparently, I didn't know this until I went to the talk, they have a research component as well. And someone there at 23andMe was interested in, of all things, susceptibility to canker sores. Canker sores is just another name for common oral ulcers. And so they, just when people would send their samples to them, there was a questionnaire that went along with it. And one of the questions was, do you get frequent canker sores, yes or no? And there were 178,409 people who said, yes, they do get frequent canker sores. And there were 66,603 who said, no, they don't. They didn't do any physical exam on these people. They didn't look at their medical records. They just took the yes or no answer to the question and did a genome-wide association study. And lo and behold, they found a number of loci, actually, that are the same as the loci that we had found that are associated with susceptibility to Behçet's disease, which is really, I found very interesting that, you know, that they could do this. I mean, it was gratifying to us that they came up with, you know, some of the same answers that we had come up with in the laborious studies that we had done with Ahmet Gül in Istanbul in order to figure out susceptibility loci for Behçet's disease, that they would be the same as some of the susceptibility loci for just common canker sores. And then the folks at 23andMe collaborated with the UK Biobank and confirmed this in a larger cohort of patients. So then enter Kalpana Mantharam. Now, Kalpana is, or was, a fellow in my lab who was studying a disease called FAPA. FAPA stands for the syndrome of periodic fevers with aphthous stomatitis, pharyngitis, and cervical adenopathy, or cervical adenitis, PFAPA. And some people pronounce it FAPA, other people pronounce it PFAPA, but whichever way you pronounce it, it is the most common periodic fever syndrome in children, much more common than FMF, for example. And it is unique in the clock-like regularity of the episodes that many of the patients with FAPA have. It's certainly the most regular of any of the periodic fever syndromes in children. And so a child with FAPA will have his or her own periodicity of their episodes, which could be 29 days or 19 days or 25 days, whatever. Whatever it is, the parents can then predict months in advance when their child is going to have, you know, their next and the next and the next episode, and they will be pretty spot on in terms of when those episodes will occur. So that's pretty remarkable, and we don't usually see that in diseases like FMF or TRAPS. FAPA is unique for its dramatic initial response to corticosteroids. If you give a child with FAPA a dose of prednisone at the beginning of an attack, oftentimes it will abort the attack, although then oftentimes the spacing between the attacks will become shorter. It runs in families, but usually not in a Mendelian pattern. And children with FAPA tend to outgrow it by young adulthood, although we do in our clinic have a few patients who are adults who still have episodes of FAPA. So Kalpana asked the question, well, if there are certain susceptibility loci that are in common between Behçet's disease and common canker sores. Might they also be susceptibility loci for FAPA? And so she tested that hypothesis, and lo and behold, she was right. And these are 4 that she found that are especially strong. So the IL-12 alpha gene, which encodes the P35 subunit of interleukin 12, is associated with susceptibility to FAPA, and the odds ratio is 2.3, which is actually quite substantial. And for a complex disease, that's really pretty good in terms of an odds ratio. But she also saw for STAT4, interleukin 10, and CCR1, CCR3, that again, there was a susceptibility association there with very substantial odds ratios associated with each of them. And so that led Kalpana to propose the idea of Behçet's spectrum disorders, that there's a spectrum of diseases with common susceptibility loci, with Behçet's disease, at the severe end of the spectrum, FAPA in the middle, and recurrent oral ulcers at the mild end of the spectrum. And Kalpana looked in FAPA patients to see whether or not IL-12 positivity as measured by flow cytometry would be increased during flares, and found that in fact the CD14 monocytes from FAPA patients do express more of the P35, P40 two-chain IL-12 protein during flares than not during flares, and that's more than control individuals. And if you look at just individuals from the blood bank who have either the risk allele, 2 copies of the risk allele, or 1 copy, or no copies of the risk allele, that in fact the amount of IL-12 produced in vitro upon stimulation correlates with the number of risk alleles that that person expresses. And so Kalpana also looked at HLA associations. She, of course, replicated the HLA-B51 association in Behçet's disease with an odds ratio of 3. She found that for FAPA patients, though, there's a substantial HLA association of class 2 molecules with an odds ratio of around 2. And for recurrent aphthous ulcers, she and others have found that there's a weak association with a class 2 allele. And so the stronger HLA associations are associated with the more severe end of the spectrum. And this is just the title page of the paper that Kalpana published in the PNAS in June, describing this new concept of Behçet's spectrum diseases. And of course, the idea is that FAPA is just on the spectrum of Behçet's spectrum disorders, and that it's within this range of normal, if you will, which extends into the abnormal. Okay, so then the third story, and we're getting towards the end anyway, and this is a story that I think is really interesting. It's not exactly a story that you guys as pediatricians would perhaps immediately resonate with, but I think that from a mechanistic point of view, it's something that is certainly worth your hearing about and knowing about. And this is a story about reversing the paradigm of discovery. And the person who is responsible for this is a person who's still a fellow in my lab, a scholar in my lab, as we call them, David Beck, who is a human genetics fellow, clinical fellow in the lab. And then he's collaborated with Peter Grayson, Marcy Ferrado, Amanda Umbrello, and Patricia Hoffman in doing this work. And so the question that these guys were interested in is the question of how can we figure out the cause of autoinflammatory disease amongst more of our patients than what we have. So I have this clinic at the NIH where we have patients who have autoinflammatory diseases. That's what I study. And we have about 3,000 patients who have enrolled in this clinic overall. And we have a molecular explanation, a genetic explanation, on about 1,000 of them. So for 2,000 of them, we don't have an explanation. And, you know, we try our best to figure it out for more patients, and we've found some new diseases over the years. And so that's been good, but we still have a bunch of patients where we haven't figured it out. And so David was then thinking about, well, how can we do better? What can we do to figure out more of these patients? The usual way that we would do it amongst our 2,000 undiagnosed patients would be to say, okay, well, let's see if we can find a few of them that share something in common. You know, a certain kind of arthritis, or a certain kind of rash, or a certain periodicity of their fevers, whatever. And that then we might do exome sequencing or some other kind of genetic analysis to find some genetic factor that would be in common amongst those individuals, and that could, if we're lucky, define a new disease. That's how we usually do it. But David instead had this idea of genotype-first. And so the idea that he had was that he's interested in ubiquitination. This is a biochemical process that occurs inside cells. I already mentioned it with regard to A20, that basically ubiquitination is a very important process that regulates a number of different signaling, immunologic signaling pathways inside the cell. And we've already found some autoinflammatory diseases that are caused by mutations in ubiquitination genes. Haploinsufficiency of A20 is one of them. Otulipemia is another. So he thought that, well, we'll just make a list of all of the genes that are associated with ubiquitination, regulating ubiquitination. And he came up with a list of 841 genes. So his idea was that he was going to look at each of these 841 genes in the exomes of a bunch of undiagnosed patients. And so he had 1,477 exomes from our autoinflammatory cohort. And just to embellish things even further, he had 1,083 exomes from the NIH undiagnosed diseases cohort. So altogether, 2,560 exomes. And the idea was that he was going to take each of these 841 ubiquitination genes and look in the 2,560 exomes and see if he could find any variants that looked suspicious and variants that might define a subgroup of the patients. And so basically, he was then looking for variants in genes with a PLI of greater than 0.9. That means genes that are pretty intolerant to variation. Variants that are not present in NOMAD, which is a database of mutations in humans that has over 100,000 people in it. And these would be variants that are shared amongst cases. And so what did he find? Well, he found 3 middle-aged men who are heterozygous for mutations at position 41 of a gene called UBA1. Middle-aged men that are heterozygous for mutations at position 41 of UBA1. Now, you know, you could have various reactions to that. One reaction would be, great, he found something. Another would be, he only found 3 people. But then the third reaction, which would be actually the most appropriate, is this doesn't make sense. And the reason that it doesn't make sense, that is not obvious from what I've told you so far, but it will be once I clue you in, is that UBA1 is a gene that's encoded on the X chromosome. So of course, nearly all men have only one copy of the X chromosome. So how in the world could these 3 middle-aged men be heterozygous, in other words, have 2 different forms of a gene that's encoded on the X chromosome, where presumably they only have one copy of that gene. Doesn't make sense. And in fact, usually the sequence analysis software would throw that out as a likely sequencing error. But David Beck is a very persistent guy. And he looked at that and he said, well, there could be other explanations for that. It could be because of aneuploidy. He looked for that, that maybe the person had an extra X chromosome, but they didn't. Or it could be because of mosaicism, because in fact there was a mutation that arose after birth in the individual, just in a subset of the person's cells, and that the reason why they look heterozygous is that they really have two subpopulations of cells, one of which has the mutation and the other of which does not. So that was his hypothesis. And so he looked at that, and of course, we get the DNA from the blood, so you would think that maybe this would be going on in the blood. And for these 3 middle-aged men, what he found was that, in fact, the bone marrow cells did have 2 versions of the UBA1 gene. Wild type is shown in red here. Mutant is shown in blue. And you can see that, maybe you can see, that there's both a red peak and a blue peak there in the hematopoietic precursor cells. What's more, in the peripheral blood, there's both a red peak and a blue peak. However, if we then look at the different subpopulations of blood cells, one can see the red peak and blue peak in megakaryocytes and erythroblasts. Oops. You can see a red peak and a blue peak in the myeloid cells, in neutrophils and monocytes. But you only see a red peak in the T cells and B cells and the lymphocytes. And so, in fact, he was right. He was right that, in fact, in the peripheral blood, there's a mixture of cells, some of which have the mutations, some of which don't. And then, of course, the other question you'd be asking is, well, what about fibroblasts? What about some other kind of cell? Do fibroblasts have the mutation? No, they do not. And lymphocytes do not, but all of these myeloid cells do have the mutation. This is just frequency of mutation along the y-axis. So that was really interesting. But what I'm about to tell you is incredible. Fact, stranger than fiction. So what happened then was that we went to the hematopathology department and met with Dr. Calvo, who's the NIH hematopathologist, one of them. And she was looking at the bone marrows with us. And she said, well, gee, look, there's all of these vacuoles in the precursor cells from these patients, from all 3 of the patients that had the mutations in UBA1. And she was saying, these are quite peculiar. You don't see these very often. I know I've seen them before, but I just don't remember where. So in any case, then we came back to see her again a few days later. And she had a couple of reports in her hand, and she very ceremoniously plopped them into my hands and said, Dan, I remembered where I've seen these vacuoles before. Here's the reports of the patients. These are your patients, Dan, from 8 years ago. You ought to go back and check these patients and see if they have mutations in this gene too. So we did go back to check whether those 2 patients had UBA1 mutations. They were both from middle-aged men, and both of them had UBA1 mutations. And when we checked, both of them are somatic mutations where there's basically a mixture of cells, some with the mutation and some without the mutation. So that was really interesting. One of those patients turned out to have relapsing polychondritis. And so that led us to go to Peter Grayson and Marcy Ferrata at the NIH, who are following a cohort of patients with relapsing polychondritis. And sure enough, some of their patients with relapsing polychondritis, besides the one that we had found there, also have mutations in UBA1. One thing led to another, and we now have and published in the New England Journal, it's out online, but not yet in print, It came out actually on October 27th, the day that David Beck presented this at the American Society of Human Genetics meeting. We have 25 patients, all of them middle-aged men, all of them who are somatic mosaics for mutations in UBA1. And here is the summary of this cohort of men. You can see that the age of onset ranges from 45 to 80. This is a severe disease. 40% of these patients have died of their illness. Fever, skin involvement that looks like sweet syndrome, neutrophilic pulmonary infiltrates, chondritis, venous thromboembolism, macrocytic anemia, and bone marrow vacuoles are some of the major features. And you can see that these patients met diagnostic criteria for a number of different rheumatologic or hematologic diseases, ranging from relapsing polychondritis to Sweet syndrome to myelodysplastic syndrome, multiple myeloma, polyarteritis nodosa, giant cell arteritis. So this is neat. And I'm sorry if it's, already 10 o'clock on Friday night and I'm telling you that this is neat, but it is, because, here what David Beck did was that he took this genotype-first approach and found individuals who have mutations in this UBA1 gene. And these individuals had a whole bunch of different clinical diagnoses before, we realized that they all had mutations in the UBA1 gene. But in fact, they have a disease that's defined by mutations in the UBA1 gene, and we can sort of take them out of the cohorts of relapsing polychondritis or polyarteritis nodosa or whatever, and put them into their own category. So it's as if we're defining a new disease by starting with a gene rather than the other way around, which is what I told you we usually do, of starting with patients that have something clinically in common and then find a gene. Here, we're just going from a gene where we think it might be interesting to defining a set of patients with different clinical diagnoses that all have something in common genetically. And so we call that disease VEXAS, V-E-X-A-S, which rhymes with Texas, and it is an acronym for vacuoles E1 activating enzyme, which is what UBA1 encodes. It's X-linked, it's autoinflammatory, and it's somatic. If you look at the neutrophils from these patients and compare them with healthy neutrophils, you can see that they have they look like, this is what neutrophils look like when they're about to die. So they really look sick. They look bad. And if you look at various inflammatory gene expression profiles, you can see that in fact in the whole blood or neutrophils or monocytes, you have an inflammatory signature. You don't have that inflammatory signature in lymphocytes, which, as I mentioned, don't harbor the mutation. We think that the lymphocytes that have the mutation die off before we can ever see them. Now, in terms of this UBA1 gene, what does it encode? It encodes a protein that is an E1 enzyme. And just to briefly mention the ubiquitination process, it involves an E1 enzyme, which actually picks up a ubiquitin molecule. It transfers it to an E2 protein, which then adds it to a target protein. And these various proteins that are involved in this are called E1, E2, and E3. There's only 2 E1 enzymes, and UBA1 is a critical enzyme. It basically, it does almost all of the UBA1, all of the E1 work in the ubiquitination process. Now, what about the mutations? I told you that the mutations are all at a particular place in the UBA1 protein. And the UBA1 protein is drawn out here. It has a nuclear localization signal. It has, actually, there are mutations in another part of the protein that can cause spinal muscle muscular atrophy. But the mutations that are associated with VEXAS are at position 41. And there's a methionine at position 1, which is a start site for translation. And if you get starting there, then you do have the nuclear localization signal, and you get a long form of the protein that goes to the nucleus. If you start at position 41, that's after the nuclear localization signal, so that you have a form of the protein, that doesn't have a nuclear localization signal. It's a little bit shorter, and it goes to the cytoplasm. And what I said is that the mutations are at position 41, so that you don't get that methionine 41. So you would not necessarily get the cytoplasmic form of the protein. And this just illustrates UBA1A is the long nuclear form, UBA1B is the shorter cytoplasmic form. So then if we look to see, well, what's going on in the cells of patients, you can see here, and by the way, I know I'm 5 minutes over, I'll be done in about 5 minutes. So, and I'm happy, as I said before, to answer questions for up to an hour, hour and a half. So if you look at either control cells or patient cells, and you look at CD3 positive T cells. So the T cells, remember I said, they don't have the mutation, and you have UBA1A, a UBA1A band in the patients and the controls, and you have a UBA1B that's the cytoplasmic form in the patients and the controls. So basically, that's just telling us that the lymphocytes are normal. On the other hand, if we look at CD14 positive cells, those are the monocytes. And if you look in the controls, you can see where the A band would be, and you can see that you still get the A band. This is the nuclear form in the patients. But in the controls, here's the B band, but you don't have the B band in the patients, in their monocytes. Instead, you have a lower band, which is here, which is UBA1C. So UBA1C is actually starting the translation at the next methionine down in the protein, which happens to be at position 67. So it makes a slightly smaller protein, and it's not functionally very active. And so you get less ubiquitin in the cell, less polyubiquitin, because basically that's not a functional form of UBA1. You get more free ubiquitin in the cell, and you get evidence for activated cellular stress shown here. So then if we do this in a transfection system with 293 T cells, you can see here are the 3 different mutations at position 41, and you don't get the UBA1B band, but you do get the UBA1C band. Now, remember I told you that we thought that the UBA1C starts at position 67. So if that's the case, then we should be able to introduce a mutation into position 67, and that would make the C band go away, and it does. So that was confirmation to us that our idea was right. And so then, if we look at the functional effect of that, here, just looking at the right-hand side, this is UBA1B, this lane here. And here, this is showing that ATP is present. And ATP would then allow for the formation of this complex of E1 and ubiquitin. And you can see that for UBA1B, most of it actually has ubiquitin bound to it. In other words, this is the normal UBA1B, and it's highly charged, if you will, with ubiquitin. UBA1C, on the other hand, is uncharged. It's non-functional. It doesn't pick up a ubiquitin chain. And that's just shown here down as a histogram. UBA1A basically has a lot of activity. UBA1B has a lot of activity. UBA1C does not have a lot of activity. What's more, we developed a zebrafish model of this disease, and that's shown here. So these are zebrafish, normal zebrafish with normal amounts of neutrophils in them. If you get rid of UBA1 altogether, you don't have very many neutrophils. If you get rid of just UBA1A, it's still okay. But if you get rid of UBA1B, you get a reduced number of neutrophils. And that's just shown here. The blue is getting rid of all of UBA1. The green is getting rid of UBA1B. So in both cases, that's reducing the number of neutrophils a lot. But if you get rid of UBA1A, it doesn't have much of an effect. And in terms of the inflammatory process, that's shown on this slide. So that, for example, for TNF-alpha, if you get rid of all of UBA1, you get a lot of TNF being produced. If you get rid of just the UBA1B, you have a lot of TNF being produced. But if you get rid of UBA1A, you don't get a lot of TNF being produced. So again, that's saying that mutation at position 41 is key to developing this inflammatory process. And then this slide just shows it to you in a different way. Wild type UBA1 leads to the production of normal amounts of the purple nuclear UBA1A and normal amounts of the cytoplasmic yellow UBA1B. The mutations at position 41 don't have any effect on the nuclear UBA1A, but do have an effect leading to the production of a non-functional or very reduced in function UBA1C, shown in green. So, VEXAS, the big picture. Using a genotype-first strategy, one can define new illnesses based on genetic variants shared among patients carrying distinct clinical diagnoses. This may give rise to a new molecular taxonomy of rheumatic disease. Second, somatic variants for genes encoded on the X chromosome are probably under-recognized. In other words, a lot of times the software that are there to analyze the data throw those X chromosome mutations, somatic mutations out as being sequencing errors. And then finally, somatic mutation may account for a significant fraction of adult onset, and who knows, maybe some pediatric onset inflammatory disease. This is the title page of the paper in the New England Journal that came out online on October 27th. You can see David Beck as the first author, Peter Grayson as the last author. And there was an accompanying editorial by Efrat Levy-Lahad and Mary Claire King on the importance of somatic mutation in human disease, which puts it into perspective in a way similar to what I've tried to explain to you now. So coming back to the title of the talk, Rheumatic Disease and the Human Experience. Well, the first story I told you was that in some cases, immune variants that confer protection against infectious agents may also cause autoimmune or autoinflammatory diseases. Second, genetically complex human disease resides on a continuum, as we saw with Behçet's and aphthous ulcers and FAPA, that extends from the range of normal experience to life-threatening illness. And then finally, somatic mutation is an inevitable part of life. In fact, we know that it's very important in the pathogenesis of cancer. And what I'm telling you is that it may be very important in the pathogenesis of rheumatic diseases as well, and may lead to serious rheumatic disease. Here are the culprits that were involved in a lot of this work. I've already shown you the picture of some of these people. Of course, these photos were taken before the pandemic, when it was okay for people to get together in a group. This is my lab, and this is my clinical team. Here are a number of our collaborators that contributed to all of this work. Ahmet Gül was particularly important. He's at Istanbul University and got a lot of the blood samples for us that we used both in the studies of FMF history and also Behçet's disease. Seiza Ozen was an important collaborator. Jay Che went to her lab in order to get blood samples from a number of patients with FMF, as well as the patients that we had from the NIH. Jim Bliska is an expert on Yersinia pestis that was an important collaborator in all of these studies. And then a number of people from the NIH who also contributed enormously to this study. And then finally, a photo of the clinical center of the NIH. This is the teaching hospital of the NIH, where all of our patients are seen in the autoinflammatory cohort. And my research lab is down in the basement over here, just in case you want to know. And if you would like to email me or my colleagues with regard to referrals, certainly that would be fine. You can just email me and I can direct the referral to the right place. dan.kastner@nih.gov is my email address. And with that, I will call it to a close. I apologize to you for going 15 minutes over, but I figured since this is the end of the day, that probably that would be at least somewhat acceptable. And I hope that this has been interesting and enjoyable and perhaps a little educational to you all as well. So with that, I'll call it to a close, and I'm happy to try to answer any questions if you have them.
Thank you, Dr. Kastner, for that excellent narration of excellent deliberation about the autoinflammatory process and its vivid manifestations. There are some questions in chat box. I will read it for you.
Okay.
The first question is by Dr. Amit Rawat. I think it's a long question. Dr. Amit, please unmute yourself and ask the question directly. Dr. Amit, you can unmute. Okay.
Can you hear me?
Yeah.
Yes, indeed. Yeah.
Fantastic talk, Dr. Kastner. So I would like to know, while it is true that the pyrene, the MEFE variants, they do have irrefutably they protect against Yersinia. I'm just curious that why were they selected only in these selected populations? Why were they not? Because Yersinia has been known to affect populations across the world through ancient times. So why are these variants in such high frequency only in these Eastern Mediterranean population? Why were they not propagated along the Silk Route to other populations?
Well, that's...
and was it because of high consanguinity in these populations? Could that be one of the reasons?
It's a wonderful question. It's a great question. And in the interest of time, I didn't go into the detail that I would have in order to address that question. But now that you're asking it, we can go into that question. And that is that if you look at people, modern day people who have FMF, and you look at the haplotype that's associated with the M694V mutation or the V726A mutation, or for that matter, the M680I mutation. Those are the three major mutations that are associated with FMF. Actually, by haplotype analysis, all of the modern day people who have FMF have the same haplotype, have a haplotype that can be traced back to ancestors that lived in the Middle East. And so I think that it is probably just a coincidence that those mutations happen to arise in the Middle East, and that plague was present in the Middle East during the period of time when those mutations were already present in those populations, and so it was selected for there. You're quite right that plague is something that, you know, is a fairly worldwide infection. And so you might think that, well, one would also see these mutations being selected for in China or in Japan or wherever. But in point of fact, those mutations were not present in China and Japan at the time that plague was present. So it's the combination, and of course, this is oftentimes the case that we're having to think about gene-environment interactions that involve a number of factors that are interacting, and so we have the interaction between pyrin and Yersinia pestis that I tried to explain to you, but also one has the fact that the mutations just didn't happen to arise everywhere in the world. Now of course, in the Ashkenazi Jewish population, which is a population that eventually did migrate to Eastern Europe, there is a high frequency of the V726A mutation. And so that is an example of where the mutation is more common in another part of the world. But the V726A, the haplotype that's associated with the V726A is the same haplotype as what you can see in the Druze population, actually, in the Middle East. The Druze are a religious group that live in the mountains of Lebanon and other places around Israel. I visited some Druze villages back in the late 1980s when I was doing the FMF positional cloning project, and the Druze have been isolated for the last 1,000 years at least. It's a very isolated society, but yet the Druze have the very same haplotype associated with V726A as do Ashkenazi Jewish people. So it's an interesting thing where there is this history that appears to be associated with it as well.
Okay, sir. Dr. Amita Agarwal madam is also willing to ask some questions. So,
okay, so Dan, I had the same question. This was a wonderful talk, and I had the same question that why has these findings been replicated in any other population where plague was prevalent? That you have answered. So corollary to that, has any other inflammasome gene-related disease selection has been linked to any other microbe? Like here we have shown for the pyrin gene. So would it be applicable to other genes in the inflammasome pathway that they have been selected because of other microbe?
Yeah, that's an excellent question. So, so far, we don't know of any other. And, I don't think that one can say that mutations in any of the other inflammasome proteins are all that common in any population. You look at NLRP3, which of course is sort of the most famous inflammasome, if you will, but the diseases that are associated with NLRP3 mutations, familial cold autoinflammatory syndrome, Muckle-Wells syndrome, and NOMID, those are all pretty rare diseases. And so, there hasn't apparently been any selection associated with them. Of course, the other thing is that you have to find a disease that, is just right in terms of being a disease that could still allow people to live long enough that they would have the disease, or a disease that looks recessively inherited too, because basically what works the best is if you have people who are heterozygous carriers have a selective advantage, and people who have 2 copies of the gene have the disease, which may in some cases be lethal. For NLRP3, 1 copy of the mutation is sufficient to cause disease, and at least for a disease like NOMID, it's a very severe early onset disease where you wouldn't survive very long with that. So FMF is special, in the sense that you have this apparent recessive inheritance. The reason I say apparent recessive inheritance is that, in fact, there are some people that have 1 copy of the mutation that can have clinical symptoms, but most people with FMF do have 2 copies of the mutation. So it does behave more or less as a recessive. And these selective advantage things work best for the recessive traits. So another famous example that's not inflammasome related would be sickle cell. So sickle cell, the carriers for the hemoglobin S mutation have resistance to malaria. And if you have 2 copies of the hemoglobin S mutation, then you have sickle cell anemia, which up until recent times could lead to very early death. But so why would you have selection for it? And it's the same process as with FMF. The carriers far outnumber the people who are affected. Because let us just say, as a simple example, an easy example to think about, that the carrier frequency in a population, this is just for example, let's say the carrier frequency is 20% in a population. Okay, so that's a high carrier frequency, as I was saying before. If the carrier frequency is 20%, the allele frequency, just by math, is half of that. It's 10%. And then the affected frequency is the square of the allele frequency. So if the allele frequency is 10%, then the affected frequency is 1%. So you've got, in this example, 20% of the population are carriers and would be protected against whatever disease it is that we're thinking about. 1% of the population would be homozygous, or 2 copies of the mutation, and would have some disease that would be lethal to them. So it's a good bargain in terms of evolution. The population loses 1% of people who have a terrible disease, but 20% of the population are protected against some bad infection. So that's why these things happen, is that if you have a disease that's recessive. And so, if you think about the inflammasome diseases, NLRP3, those are dominantly inherited diseases. NLRC4, also, it's usually de novo and dominantly inherited. So I think that's what we're dealing with too. So there's special circumstances that have to be present in order for this to work.
Okay, sir.
Yeah.
Dr. Suma, please put your...
Yeah. Hello, Dan, that was a fantastic talk. I really enjoyed it, especially the last recount of moving from genotype to diagnosis. That was very interesting. Actually, my question was, why isn't HLA-B51 in the diagnostic criteria for Behçet's? Because, I mean, I live in Kerala, and we are on the old Silk Route. And we do get to see quite a few patients with, forme fruste of Behçet's. And while you get to see some patients who actually, childhood pediatric Behçet's as well, while you tend to see, some children who can actually fulfill any of the diagnostic criteria, there are several children I see who have maybe one major component and a B51 to go with it. They do not have enough criteria to actually make a diagnosis. And the question is, what relevance do I give the B51 here? So I have children who come with severe visual threatened retinal vasculitis, bilateral, and no other features whatsoever. And they've got a B51. I had a 1-year-old infant with severe aphthous ulcers, oral ulcers. I mean, very significant, high frequency occurring oral ulcers, who was B51. Then I have 2 children who actually presented with PO, pyrexia of unknown origin, and on investigation, they actually had aortic dilatation and thickening of the aortic wall, who both have a B51. And so the question is, what label should you give these people, and how do we explain? I mean, how do we actually characterize these kind of diagnoses?
That's a wonderful question. And, you know, I think that you're really getting at what Kalpana is getting at with regard to there being a spectrum of disease that is in this Behçet spectrum. And you know, for the purposes of the criteria, you know, like the ISG criteria, for example, for Behçet's disease, those criteria were developed, at least to some extent, you know, so that clinical trials could be done and you would know that, you know, all of the patients that were in one group, you know, met criteria or whatever. And the criteria don't necessarily always work so well, you know, when you're on the front lines as a clinician and having to think about what's going on with the patient. And I guess that Kalpana's point, or a major point that she's making, is that really this is a spectrum of disease, and that you have to think about the biology here, and that, you know, there are probably a number of genes that are involved, and maybe eventually we will redefine things. You know, we'll use a different nomenclature rather than just saying that somebody either has Behçet's or not. You know, that maybe we will recognize Behçet's spectrum disorder. I will say that, you know, with regard to your question of B51, B51 is not the only HLA class 1 that is associated with Behçet's, and there are several others. B15 is one of them. I don't remember them all, to be honest with you, but you know, in that Mike Umbrello paper from 2014, it goes through all of them that are associated, or there's even some that are protective. So, you know, I would, you know, for your patients that you're talking about, you know, like a patient that's B51 positive and has vision-threatening uveitis, if I were you, and of course, you know, part of the issue is it available, but if you could, I probably would be treating that patient with a TNF inhibitor, you know, in the hope of preventing visual loss in that patient, because it's probably the same pathophysiology, even though the patient doesn't meet, you know, the ISG criteria for Behçet's disease, but it's probably the same biological process. We have found actually that, you know, for the oral ulcers, we see, you know, we're sort of a referral center for a bunch of different kinds of things. And so both kids and adults who have very, very severe oral ulcers oftentimes get sent to us. And at least some of them do respond to TNF inhibitors as well. Apremilast is another agent that, you know, sometimes, at least for the oral ulcers, works reasonably well in these patients. And we've found that apremilast is an inhibitor, among other things. It's an inhibitor of IL-12, you know, which Kalpana found to be an important susceptibility locus, you know, for this Behçet spectrum disease. So I think that we have to think a little bit more broadly about these kinds of things, and I think that the classification system will evolve, you know, as we understand a little bit more about these things too.
Thank you.
CK?
Thank you.
Thank you, sir. CK, sir. Sir, please unmute yourself. Sir, unmute yourself, sir.
Yes. Can you hear?
Yeah. Okay. Now,
can you hear me?
Yeah,
yes, indeed.
It's okay.
Yeah, I again want to talk about intestinal Behçet's we had very recently. A 8-year-old child who had all the inflammatory markers, and he suddenly went into intestinal perforation. Once operated, he had 9 perforations in the small intestine. We had to forego the almost 30 centimeters of intestine. We had to do a ileostomy, and he was reoperated 3 days back. Now the point is, he had only 1 ulcer in the gum, only 1 ulcer, and high fever. No other markers, no CNS manifestations, no genital lesions, no eye problem, nothing he had. So only the intestinal manifestation, very, very unusual. And we could make a diagnosis by the histopathology examinations. That is one. Second question I would like to ask you is, do you recommend the use of chloroquine in children coming with rheumatic valvulitis? Because it's a problem in developing countries. Whatever precaution you take, then you continue to them, they go for deranged the valvular damage and get it lifelong problem for them. Suppose, as it is being recognized now, the use of chloroquine, how long you should use it? What's the dose you recommend in those situations? Thank you.
All right. Well, so the first question of, the connection wasn't real good, but I take it that this was a child who had as the only manifestation was intussusception and intestinal perforations. And the question is, what's going on in that patient? And you know, I would be very interested if you were able to provide it in doing DNA sequencing on that patient and the patient's parents, because there could be a number of different mutations and different genes that could lead to that. And so it could be, for example, a gene that's in the IL-10 pathway that could be involved in these kinds of processes. And so, you know, if there's some way that we could arrange for you to send DNA specimens to us, or you can do it in India. You know, if you have someone that wants to do it there, that's totally okay with me. But we'd be happy to do that for you, to try to figure that out and see if there could be a molecular explanation. Because a lot of times when you have these kinds of unusual presentations, you know, where it's just intestinal, for example, you know, it could be
already I'm in contact with Dr. Geeta on this issue. She has collected the samples for the sequencing, and the HLA-B5 in this boy was negative.
What was negative? I'm sorry, I didn't hear what was negative. Something was negative, but I didn't hear what it was.
I guess some connection problem is there.
Yeah, but you know, I'd be happy if we can't reestablish the connection to do an email with him afterwards.
This is the last question, sir. This is from Dr. Sumita Danda. Will this somatic mutation in UBA1 be detectable in peripheral blood DNA?
Absolutely, yes, it is. At least so far, the people that we've seen who have it are all middle-aged elderly men. But at this point, you know, the paper had 25 patients in it, I mentioned, and you know, there's a table of them in the paper. But we're now aware of 70 patients with this, just, you know, since we started talking about it. So this is going to turn out to be a fairly common thing, I think, at least in adults. So yes, it is detectable in peripheral blood, and the variant allele frequency, at least amongst the patients that we've seen, is pretty high. You know, it's like 50%. So it's not like, you know, only 1% or 2% or something like that of the cells in the peripheral blood have the mutation. It's a lot of them, at least in these patients that have clinical findings. It's quite a, now of course, you know, it must start out as a low percentage, and it may be that eventually, you know, we will find patients that have low percentages, and we're looking for them now, but at least so far, all of the patients that we've seen with it have had pretty high variant allele fractions.
Okay, sir. Dr. Kishore?
Oh, hi, Prof. Kastner. It was an absolute
pleasure. You're audible, Kishore.
Am I audible?
Yeah, you are audible.
Yeah, Prof. Kastner, it was an absolute pleasure to kind of listen to you again. I had just listened to your talk at press. It was so uplifting for us. And then I've met you a couple of times at the
Okay,
ESAID meetings. I work with Elizabeth McDermott. We do our auto-inflammatory clinics together in Nottingham. But as
Yes.
always, it's been an absolute pleasure. We've met a few times, but and you've been very kind with your email queries, and you've always been very prompt in responding and stuff. But I think that was an inspiring presentation. Thank you very much.
Well, that's very kind of you to say, and I really, really appreciate that. And Elizabeth McDermott, gee, she goes back to TRAPS days with us in the UK.
We still have our
Nottingham.
big cohort, and I think new members of that family are getting diagnosed almost by the day. There are 2 more who were diagnosed in the last year who are under my care.
Wow! Wow!
Thank you.
Well, that's
Kishore.
fantastic. Fantastic.
Okay. Dr. Geeta Govindaraj, the chief coordinator of this program, also put some questions in the chat box. I request her to ask it directly, and also request her to conclude the session.
Okay, thank you, Professor Kastner. That was a fantastic presentation. Just 2 very simple questions.
Okay.
Do we really now know for sure what causes this regular periodicity? So some patients often tell us that it's just like menstrual periods. So what exactly underlies this exact periodicity in a few of these recurrent fever syndromes like PFAPA and cyclic neutropenia? And the second question is, why do some of these autoinflammatory syndromes get better with age, like hyper IgD syndrome, and some of them, like TRAPS, seem to get worse with age? Do we have an answer? Very simple questions.
Yeah, well, there's simple questions to ask, not so simple to answer. I guess it would be what I would say with regard to that. So the first question with regard to the periodicity and how can it be that some of the episodes, at least for some patients, that the episodes are very, very predictable in terms of their periodicity, like for PFAPA and perhaps other diseases as well. And I must say that we don't have a good explanation for that. Kalpana and I have talked a lot about the periodicity of PFAPA, and she has a theory about this that has to do with the fact that one of the subunits of IL-12 is also a subunit of IL-35, and it may be that what one sees is something where, at least for part of the cycle, that the subunit is associated with the other subunit of IL-12, and then at some point it alternates to the other subunit of IL-35, which is a suppressive cytokine. And she's worked on that a little bit, but at this point doesn't have enough data to say for sure. But it could be that a year from now or 2 years from now, Kalpana will have the answer for you. And she's working on it, as one of the things that she's working on anyway. So that's an interesting question. And then you raised the question of, at least for some women of childbearing age, that the episodes can in some way coincide with the menstrual cycle. And yeah, I mean, in some way there is a connection there, and it used to be, before colchicine was recognized as a treatment for FMF, that women with severe attacks of FMF would be put on oral contraceptives as a way of trying to sort of even out the hormonal milieu. But again, we don't really understand that very well. And, we thought that we were going to be so smart. That's always the way, is that you think you're so smart, and then you learn that you're not actually. So we made these knock-in mice, that have FMF mutations. And the knock-in mice, they do have inflammation, no question about that. But it's not periodic. And so it didn't help us at all. We thought that with the mice that, we could then do various studies that you couldn't do in humans. And in fact, the fevers are not periodic. Now, of course, mice don't have a menstrual cycle. You know, they have an estrus cycle, and so maybe that's why the mice don't have you know, periodicity of their fevers. I just thought of this as I was answering your question. Maybe that's the reason why the mice don't have periodicity. I don't know, but you know, that's the kind of question that you might try to answer with an you know, an animal model. So that's one thing. And then, you know, the question of, well, why do some patients outgrow the episodes with age? For hyper IgD syndrome, I don't know. For FAPA, you know, where again, patients do seem to, most of them do outgrow it. It, I think, has something to do with the immature immune system, but what exactly it has to do with the immature immune system, we don't know. Again, Kalpana is working on that. She's gotten tonsils from a lot of patients who have FAPA, and then tonsils from kids that just have a tonsillectomy for some other reason as well. And she's found some differences in, you know, like the histology of the tonsils and so forth. And so at some point she may figure out, you know, what is so peculiar about the immature immune system and why you would get these episodes. But again, we don't have a good explanation. Great questions, lousy answers, at least right now.
Geeta, before your concluding remarks, Amita madam wants to raise an issue.
Yeah, I just had two more questions, Dan, for you. One is that you have canker sore, PFAPA, and Behçet's as a spectrum. But in the first two, we have a HLA class 2 association, while in Behçet's, we have a class 1 association. So why do we think that the HLA association is different if we think them as a spectrum? Is it that the antigen which is linked with these diseases is different, or is the environmental trigger is going to be different? And the canker sore and PFAPA are very milder disease. And in Behçet's, we have HLA class 1 association, and we have something else which is interacting and causing the disease to be more severe. That's regarding the Behçet's. And the second is regarding your VEXAS. In that, because I presented that journal club today morning in our department from the Nature NEGM. So I was very perplexed that why do we have vacuolation? I mean, what leads to that vacuolation?
Okay, so your first question with regard to HLA and the Behçet spectrum disorders. So that's an excellent question. And, you know, when Kalpana and I were talking about whether to propose this idea of the Behçet spectrum disorders, it was with the idea that the spectrum was defined by the non-HLA genes, you know, by things like IL-12 and STAT4, and that those are what define the spectrum, and then what determines where you are on the spectrum would be the HLA, you know, as to whether it's class 1 or class 2. And you're right that the class 1 or class 2 probably does, to some extent, determine what antigens are important in the pathogenesis of that. So I definitely agree with that. And I think that the thing to keep in mind is that the spectrum is really defined by the non-HLA rather than the HLA. Now, with regard to the VEXAS question of what are those vacuoles? What do they have in them? We don't know. I mean, that was one of the questions that we were interested in, you know, from the very beginning. And David Beck actually has stained the cells from patients with VEXAS with antibodies against various things, you know, and has used immunohistochemical stains, you know, that will pick up this or that, you know, chemical inside the cell. And so far, there's not anything that has come out as being the, you know, what it is. It could be just that, you know, these cells are undergoing cell death and that it's a bunch of junk, you know, that's in there, you know, that the cells are undergoing autophagy and that it's not any one thing in particular. We don't know. It would be really interesting to find out, though. I can't hear you. You're muted.
Is anybody asking a question?
No, I'll just. Thanks a lot, Dan, for a wonderful lecture and such a good explanation for all the questions.
Oh, well, thank you. Thank you. That's very kind. All right, other questions that people may have?
Dr. Vinod has done some work on familial Mediterranean fever. Dr. Vinod and Dr. Sandhya have published articles on familial Mediterranean fever from India. Do you have any questions, Dr. Vinod?
No, I don't have any questions. Thank you, Dr. Kastner, for the wonderful talk.
Well, thank you for having me. I really have enjoyed this a lot.
Shall we conclude if there are no more questions?
Yeah. Ida,
Yeah, Dr. Ashraf?
please conclude. Okay, okay, conclude, please.
So, a respected guest of honor, Professor Kastner, respected teachers, colleagues, students, and friends. On behalf of the organizing team that includes the Indian Academy of Pediatrics, Calicut, the Department of Pediatrics at Government Medical College, Kozhikode, the CSIR Institute of Genomics and Integrative Biology, Delhi, the Guardian Consortium, and the Foundation for Primary Immune Deficiency Diseases, I wish to express my deep sense of gratitude to Professor Kastner for accepting our invitation so graciously and enlightening us on a disease that is still an enigma to most clinicians in this part of the world. At Government Medical College, Kozhikode, we have been conducting a collaborative program along with the Institute of Genomics and Integrative Biology, Delhi, over the last 5 years on primary immune deficiency disorders. This was supported by the Science and Engineering Research Board, Delhi, FPID, and the CSIR. As a result, we now have a fairly large cohort of children with autoinflammatory syndromes. And in fact, we also have the largest cohort of the hyper IgD syndrome in the country. This has been possible through application of NGS technology made available free of cost to our patients through the Guardian Consortium, which is a large research network of clinicians and scientists in the country. We feel that increasing awareness among clinicians, especially among pediatricians, is a critical cog in the wheel for the recognition, and management of these autoinflammatory syndromes. This talk would definitely go a long way, I feel, towards restoring the happy childhood to affected children, while at the same time mitigating the financial and psychosocial burden and distress of affected families. Today happens to be the International Child Rights Day, and I'm sure that our scientists, clinicians, and the pharmaceutical industry would come together and ensure that the availability of the much needed biologicals that we do not have now in the country, and thus ensuring that our children will not be denied the benefits of precision medicine. We hope that your session will provide the much needed impetus for a rapid sea change in the situation. I would like to thank, on behalf of the organizing team, each and every one of the delegates who have joined us from different parts of the world. We also would like to express our gratitude to Ms. Karen Tillman for her tireless work and her help over the last few weeks in arranging this talk. And we also thank Dr. Ranjit P., web editor of the Indian Academy of Pediatrics, who was our host this evening. We are really indebted to you, sir, and will always remember this talk that you gave tonight. Thank you all.
Thank you.
Well, thank you, Geeta. Thank you so much. Thank you all. I've
Thank you,
really
sir.
enjoyed
Thank
it. This has been a wonderful honor. Thank you.
Thank you so much.
Let us end the meeting. Thank you.
Okay, bye-bye.
Bye.